Due to fossil fuel burning, CO2 concentrations in the atmosphere are increasing. New strategies to remove CO2 from the atmosphere are already being tested. It's also very important that we better understand the natural processes that are already removing it. One of these processes is performed by microalgae. Microalgae fix as much CO2 from the atmosphere as all plants on land. They create about 45 gigatons of organic carbon per year. 45 gigatons correspond to the weight of 300 million blue whales. Most of this organic carbon is fixed in the form of sugars, also known as polysaccharides. The majority of these polysaccharides are eaten by bacteria and other heterotrophs, and then the CO2 is released back into the atmosphere. However, some of the polysaccharides escape degradation, but we don't know which ones. In my research, I study microalgal polysaccharides. These polysaccharides are part of the microalgal cell, but some of them are also secreted into the surrounding seawater. From previous studies, we know that some of these secreted polysaccharides assemble into small particles that are known as transparent exopolymer particles, or TEP. These small particles are sticky and they act as a glue between microalgal cells, minerals, and other molecules, promoting aggregation and formation of bigger particles. These bigger particles are denser and sink. They are known as marine snow because they resemble snowflakes falling from the upper layers of the ocean. They sink and export carbon to the deep ocean, where carbon can be stored for thousands of years. It is this sequestered carbon that is responsible for ultimately lowering atmospheric CO2. This transport of microalgal organic matter to the deep ocean by sinking particles is known as the biological carbon pump. Although polysaccharides are important in the carbon cycle, we know very little about which different polysaccharides are produced by microalgae. In order to investigate their role in the carbon cycle, first we need to identify them. Our hypothesis is that certain microalgal polysaccharides are more stable than others. Specifically, polysaccharides that have very complex chemical structures may be more stable compared to polysaccharides with simpler structures, as the ones with complex structures are more difficult to degrade by bacteria and other heterotrophs, because they require more enzymes to break them down. And therefore, these more stable polysaccharides can accumulate for longer time, storing carbon in the ocean. In this study, we wanted to identify polysaccharides that escape degradation and that are involved in particle formation. In this study, we wanted to detect polysaccharides produced by microalgae. So we collected samples during a 3-month microalgae bloom at the North Sea, and then we extracted the polysaccharides from those samples. Then we needed one method to detect polysaccharides, but these polysaccharides are really difficult to analyze because they have extremely diverse and complex structures. They are made from monosaccharides that can be connected with different linkages. They can have side chains, they can have further decorations like sulfation. They are very challenging to analyze with currently used methods like NMR or mass spectrometry, especially when you have samples that have a mixture of polysaccharides. It's very difficult to distinguish them. Here, we used a novel approach by combining carbohydrate microarrays and monoclonal antibodies. With carbohydrate microarrays, we could analyze many samples in parallel, and monoclonal antibodies are very specific. Each monoclonal antibody recognizes one specific epitope. An epitope is a particular part of a polysaccharide structure. So we have, for example, antibodies that recognize beta-1,3-glucan, another would recognize beta-1,4-mannan, and so on. If in our sample there are many different polysaccharides together, the antibody would only bind if its recognized polysaccharide is present. Second, we wanted to know if some of the polysaccharides were present in these small sticky particles. And for that, we used fluorescent microscopy combined with monoclonal antibodies that were labeled with a fluorescent tag. Third, we wanted to know if some of the polysaccharides could be degraded by bacteria that were present during the microalgal bloom. For that, we studied the bacteria by DNA and protein sequencing, and we focus our analysis in enzymes that can degrade polysaccharides. We found 27 polysaccharide epitopes, and we followed their abundance during the course of the microalgal bloom. The most interesting finding was that one of these polysaccharides, which is known as sulfated fucan, was accumulating. The sulfated fucan was until now only known to be produced by brown macroalgae, but we found that microalgae also produce it. Sulfated fucan has a complex structure with an alpha fucan backbone that is extensively sulfated and can have side chains with fucose, galactose, and other sugars. Previous studies have shown that sulfated fucan from brown microalgae is difficult to degrade by marine microbes. Because of its complex structure, sulfated fucan degradation requires a vast collection of sulfatases and other carbohydrate-active enzymes that impose a high energy investment on bacteria. While degradation of, for example, laminorin, a sugar with simple structure, requires only 3 enzymes. We found that the microalgal sulfated fucan was accumulating during the bloom. This indicates that bacteria were not degrading it. Also, our proteomics data showed that bacteria were expressing far less enzymes for the degradation of fucan than for other sugars like laminorin. So, in this study, we found that sulfated fucan was storing carbon, was acting as a carbon sink, at least during the bloom. In addition, we were very excited when we saw where this sulfated fucan was localized. We could see that sulfated fucan was located around the microalgal cells, coating the cell, but we also found it in small particles that resemble these small sticky particles, suggesting that they could promote the formation of bigger particles. We could show this by our microscopy studies. In addition, our microarray data showed interesting abundance dynamics, where concentration of sulfated fucan increased in the dissolved fraction at the beginning of the bloom and then decreased to a constant abundance, while in the particulate fraction, it increased from the middle of the bloom until the end. These dynamics indicate polymer assembly. Therefore, our results showed assembly of dissolved sulfated fucan into small step sticky particles. Again, this suggesting that this can promote the formation of bigger particles. If sulfated fucan is resistant to bacterial degradation and forms these sticky particles, it means that the sticky glue will remain. So the particle will hold together and will be able to sink to the deep ocean where its carbon content can be sequestered for thousands of years. We found a molecule that has a rate of synthesis by microalgae higher than the rate of degradation by heterotrophs, and this is what we need for carbon storage in the ocean. Sulfated fucan is the first molecule that fulfills this requirement. But why do microalgae produce this molecule? Sulfated fucan is resistant to bacterial degradation, so we think that microalgae coat themselves with this molecule because then it acts as a barrier against bacteria and their digestive enzymes. But why do microalgae secrete also some of this molecule into the surrounding seawater? Because those ones will end up making sticky particles that will make the microalgae aggregate and sink. And at least at the beginning of the bloom, microalgae should be on the surface of the ocean where sunlight will allow them to grow. So it's not clear why they secrete this polysaccharide. Also, maybe microalgae are secreting more of this sulfated fucan because of the current high concentrations of CO2 that we have in the atmosphere, or maybe they are producing less. We don't know, but now we can investigate it because we found this molecule and now we can monitor it. Our data can also be used for blue carbon strategies. For example, if new projects want to focus on microalgae, they could check which microalgae species secrete more of this sulfated fucan, and they could go for these species and cultivate them to store carbon. We studied polysaccharides that were present in the sea surface and found that sulfated fucan accumulated and was forming sticky particles. Next step would be to analyze if this sulfated fucan is transported and make it all the way into the deep ocean. We have already analyzed it, and we just finished an article showing that sulfated fucan was present in deep sea sediment from the Red Sea. With these two studies, we demonstrate that sulfated fucan is a good molecule for carbon sequestration in the ocean. We are now analyzing several cores, and we are interested in knowing if sulfated fucan is present in marine sediments from different locations of the globe. Taking a step further would be to investigate how much sulfated fucan is produced and how much is exported. We need numbers to understand the importance of this molecule in the global carbon cycle. Also, in theory, all what is fixed by microalgae could be released by heterotrophs. We need to understand how microalgae store carbon in the ocean. Further studies with high molecular resolution are needed to understand the role and importance of the different microalgal polysaccharides in the biological carbon pump.